So our next speaker is F.Ozlugedik from Helmholtz Munich, and he will be presenting HematoGraph, a graph-aware hierarchical pooling for cell-level hematology classification. Welcome.
How should I switch? Okay. Hello, everybody. Okay. Imagine that you are sick and you do not feel well for a week, and then you visit the doctor. And the doctor takes your blood and visualize under the microscope. And this is approximately how it seems. Based on the existence of a specific pathological cells, or based on the change in the distribution of the cell lines, the doctor decides if you have the disease or not, or if you have the disease, what is the most likely thing, or what is the correct next step. Depending on the cells which is seen in the blood smear, you can go under the chemotherapy, or for the further diagnosis, you can be referred to bone marrow biopsy, which is an invasive process. In our lab, one of the main questions we want to investigate is how can we automate this process with the help of computer vision and artificial intelligence. To do so, we have three-stage pipeline. First of all, we extract 500 white blood cells across the perfect blood smear, which is the World Health Organization suggested number of cells. And then we encode these cells with a foundation model. But this foundation model is not a dyno or clip, which is the natural image trained. It's a foundation model specific to the single cell image, which is the dyno blue. And then we get 500 vectors which have fixed dimension, and we use these vectors for all of our downstream tasks. As I mentioned, the doctor decides based on what he sees across the whole slide, and the decision is in the patient level. Therefore, we have to aggregate these individual vectors to a patient-level vector. To do so, the simplest thing is to just take the mean, but it's not the optimal one, because the pathological cells can be hidden into a lot of redundant health cells, or the thing that decide the diagnosis could be the distribution, not the existence of the cells. Therefore, the community uses learned functions to aggregate these individual embeddings. It could be an attention-based pooling, where one network scores each embedding, or you can use a transformer and take the class token. And then you can also calculate the attentions of the individual patch, which helps you to quantify the interpretability or how correct the model is. Then after you get your patient-level token, you want to classify it. You can use a linear layer or MLP to map into the dimension of your labels, and then you get a prediction in the patient level, how likely this patient is acute leukemia or MDS or healthy. And then you have the ground truth in the patient level also during the training time, and you can calculate your loss function and backpropagate end-to-end the classifier and the aggregator. Yeah, this is the summary of the whole pipeline. When you calculate the predictions, you compare with the ground truth and backpropagate to train classifier and aggregator. In most of the cases, the patch encoder, the dyno blue in that case, is frozen. Okay, I want to show off two real-world examples that was done in our lab before. One of them is the AML, acute myeloid leukemia subtype classification. It was done by attention pooling. Yeah, the features are obtained, and then each features are scored by another network to obtain the attention scores. Then the attentions are multiplied with the features, and then final patient-level vector is given to the classifier. And as you can see in the right side, the confusion matrix is pretty good. And what if you want to classify all of the blood diseases, not only the acute myeloid leukemia? Then this is the idea of the cytomorph paper that was published in this year. And here we used transformer as aggregator, and then we obtained a classification for much broader range of malignancies. And it can be used as triage tool in the real world. Okay, this aggregation is something we have to improve because this is one of the bottleneck in this process. One idea we also proposed is to use mixture of aggregator instead of a simple aggregator, and during the test time, we can dynamically select which aggregator is the most useful. However, any of these ideas do not model the cell-cell interaction. Because in theory, in transformer, you can model because you have to calculate self-attention between the all pairs. But there are a lot of redundant healthy cells. Therefore, the real cell-cell interaction for the disease is not really modeled. The best data structure to model this is to use graph and the graph neural network. Actually, the graph neural networks have been explored widely in pathology. They patchify the image, and then they built the graph based on the special connections between the patches. But in hematology, we do not have this special relationship, because when you give your blood, it's just a mixture. There isn't any special location on the slide. Therefore, you cannot build such a graph similar to the right side very simply. Therefore, you have to also learn the graph end-to-end if you want to use graph-based classification or graph-based aggregation. This is our main contribution in this project. We proposed end-to-end learning for graph, and then we use graph neural network, the message passing, and then aggregation again with graph convolutional network, and then classify patient-level vector. Basically, this whole pipeline is or it enters directly into our main scheme from this aggregation step. Yeah, this DynaBlue encodes each patch, and then graph is learned, and then on the graph, the aggregation is done. Okay, how did we learn the graph? It's not a very simple task because you have to learn the adjacency between each cell pair. We map our embeddings to another auxiliary space, which is learned also end-to-end with the training loss. And in this auxiliary space, we calculate distance between each pair. And then, in this space, we sample a graph with type K Gumbel softmax, and built for k equals 10, we built a KNN graph. But important thing is the KNN graph is defined in a metric space, which is learned end-to-end. Therefore, the graph topology is also learned end-to-end. And in this space, we use hyperbolic distance because the cell hematopoiesis is something hierarchical, and this hyperbolic geometry is the best mathematical model to store this inductive bias. Okay, and then we did message passing, and then we did the aggregation. Okay, then the results. We compared our methods with almost all of the fundamental aggregation methods across the computational pathology, and we showed that there is a consistent improvement across the hematology data sets. All of them are patient-level hematology data sets, and even compared to the whole transformer, there is a considerable amount of improvement in the balanced accuracy and weighted F1, and this improvement is consistent across 4 different data sets. It's important to note that 4 different data sets have different origins. And we did an ablation study and showed that all of our choices have contribution to the final performance. But there's one thing I want to mention. When we change the distance to Euclidean distance, there's a significant performance drop. It shows that using correct inductive bias helps a lot for the final performance. And this is a GNN explainer result run on our learned graphs. And actually, it has very good results because, for example, for the first class, this mutation type, CBFB, is related to monocytic myoblastic leukemia, which means the monocytes are also high. And it shows that the monocytes are essential for the classification. And the same thing is true also for the PML rara, which is a promyelocytic leukemia. it shows that, yeah, these graph-based methods learn useful and real structure from the data. And yeah, the final takeaway is multiple instance learning is useful for hematological disease classification, and graph is correct or one of the correct inductive bias that can be used. This is the end of my presentation. I want to thank to my supervisor, customer, and Furkan Taşlayan, he's also my supervisor, and all members of my lab. Thank you.
Thank you for this nice presentation. We have time for a question. Okay, then maybe I was wondering, and you kind of showed it maybe in the last analysis slide, what kind of biological or morphological relationships does this latent graph learn, like between these different cells and so on? Have you looked into this?
I actually,
No, can you speak? Can you speak in here?
Okay. The latent graph learns between which cells there should be a message passing at the end. And it doesn't learn something directly to the cell morphology because it learns the connections of the embeddings.
Yes, but obviously I was wondering the embeddings, they must represent something.
Yeah, exactly.
Like something about
Yeah.
the cells.
For example, in this final figure I showed, for the CBFB leukemia, the monocytes are the most important cells to decide. It implies that I think it learns the correct cell group as the centerpiece of the classification.
I see. I see. Cool. Fascinating. Okay. Sorry, I think in the interest of time we need to move on, but you can, I think, catch all the speakers in the coffee break as well. Thank you again for the presentation.